All right, good morning, everyone. It's great to be back. I enjoy coming to workshops like this and getting to talk about science some. I get to, as you know, chief scientist, it's more like chief scientist administrator in the day-to-day right now, but so it's great to be here, and I am going to be able to stick around this morning for some of the breakout sessions afterwards. So I gave a talk last year at this conference. It had the same title. So I talked about why do we need to move new science into the drug development process? How do we translate new drug development tools into regulatory use? And what specific relative regulatory questions can new drug development tools address today? So yeah, that was my talk from last year. I think I can, I'll do a little more though. And so we talking about why, also covered in the 2023 version last year, how advances in technology, systems biology, stem cells, engineered tissues, mathematical modeling present new opportunities to improve our ability to predict risk and efficacy. We have microphysiological systems, other complex in vitro models, which is a big focus of this workshop, obviously. Opportunities for combined in vitro and in silico models. I think there's a lot of opportunity here. Also, genetically engineered cellular models. We've had applications at FDA where we've used genetically engineered cellular models to approve drugs and expand the indications for certain rare diseases where it's very difficult to run clinical trials. And these advances can help bring products to market faster with improved efficacy and decreased risk, toxicological or safety risk. And that's the goal. I think there's a lot of promise of these new technologies. That's one why. And another promising technology. So last week, I was at a different conference. It was actually out in Little Rock, Arkansas, where the National Center for Toxicological Research is. It's part of the Office of the Chief Scientist at FDA. And the commissioner and I both went out there, and he gave the opening remarks, and I gave some closing remarks at this conference. We actually had good attendance of people coming to Little Rock, Arkansas, which was great. And it focused on it was the Global Symposium of Regulatory Science. The title of the conference was Digital Transformation in Regulatory Science. And there were a lot of AI themes. It was, is regulatory science ready for artificial intelligence? There were some debates about this. And so I was trying to figure out what to do for the closing remarks. And I decided to take the conference program during the conference and input it into an AI image generation software and ask the software, the AI software, to make a summary image of the conference. And this is what we got. I could never have come up with this myself. So you can see we have a global transformation, all sorts of things popping out of the globe, digital transformation. We got the acronym. It's in Arkansas at the bottom. If you read, it says digital transformation in regulatorial science. So it very creative spelling. It was not within the context of use of this model. And I did look into this a little further. And yeah, of course, the large language models are trained and they spell very well in every language. But the image generation software models, spelling isn't their forte. So, you have to understand the use of a model. Doesn't mean this can't be useful, but if you understand the context of use and the limitations and how you would use it. So then I thought this week I said, make a summary image of my talk from last year at this workshop. And I fed it into my slides, and you can even just go to YouTube and ask it to summarize that way too. And this is what it came up with. And it was a little crazy, I thought. I think it did better with the AI conference image generation. There is a lot going on here. And yeah, I mean, this was, yeah, feeding in my slides from last year. We got what, how, and why, and what, and the spelling is not good. And the word it apparently struggles the most with, if you look in the upper right, is regulatory. Now we got regulatail. So, I think the AI models do not like regulation. And I like how in the middle left it says systems biology what? Like, I don't know. I historically have been quite skeptical of black box models and like to understand how things work, which is kind of what systems biology is. So I don't know. It's giving us some messages here, I think. And but I tried something simpler. We'll get to this workshop focusing on complex in vitro models. Create an image of an organ on a chip. And oh, this was pretty good, I thought. This is quite impressive. We got some injection needles and IV drugs. But, I went to medical school. I haven't practiced medicine since then. And I skipped some days in anatomy lab. But I haven't seen, I may have missed this organ. It is a strange organ, some compilation of different organs. So I tried, how about just a liver on a chip? And this is what we got. That was pretty impressive, I thought. It looks like a liver. The spelling, don't look too close. Just appreciate the creativity. And we focused on liver on a chip last year at this workshop. And then this year, I know I wasn't here yesterday, but we've been focusing on some other organ systems and applications. So I was having fun. I tried a few more. Heart on a chip, pretty good. I like hearts. Kidney on a chip. Nice cross section of a kidney. This creativity, a brain on a chip. This is my favorite. That we got neurons as the computer architecture, and we got IV tubing going into the chip. I mean, this is it's doing pretty well without no words on this slide, so that's good. Oh no, this is my favorite. Hopefully, no one's eating still. We got intestine on a chip. Oh boy. So again, don't look too closely at the spelling, but we have some creativity. I think this was my last one. How about we interconnect it with artificial intelligence, and this is what it came up with. And oh, one more, the speeding drug development. I asked it to come up with something speeding drug development. This one may look familiar. I actually put this on the cover slide. And yeah, I knew I liked hearts. It has a heart clock in the middle and pink sand speeding up drug development, apparently. It's pretty fascinating, I think. And again, it has some issues, but if we understand the context of use of different models, you could put restrictions around what you would use a model for. And I think that there are some good parallels to talking about drug development tools, those complex in vitro models. And so how do we translate whatever the new tools are into regulatory use? This is a modified version of a slide I presented last year. There are multiple steps required to translate these new technologies into regulatory use and maintain the same standards of safety, efficacy, quality, and performance. So context of use, that is very important. I think that's been covered. I know we covered it last year. I'm sure it was covered yesterday. And I think of this, for those that aren't familiar with context of use, as the indications for use on a drug or a device, and describes what it's indicated for, what the data supports the use of a given model or drug development tool for. And then in characterizing it, and what do we want to be able to use a tool for regulatory decision making? Well, we want it to be predictable or have predictability. We want it to be reproducible, that you can use it anywhere in the world and get the same result. You need to have quality control. These are some of the technical factors, and how good that is can then inform the context of use and what you would use a new model for. So going back to a human-generated diagram, at the top would be the sort of scope of something I could create, except it would be more stick figures. I think bringing stakeholders together is key. And no matter what the technology is, you have to get the different stakeholders together and talking in the same room. I know I found the breakout sessions last year very informative, and I never would have predicted where they would have gone. And I think what was I am very valuable there, and I imagine will be valuable here too, is that you had regulators, you had pharmaceutical industry scientists, you had developers of technologies, of the complex in vitro model technologies, academic investigators. I'm leaving some out, I'm sure. And when you have everyone at the table, you hear different perspectives. It's very important that, for example, if you're a developer of a new technology, it can't just be that FDA says, oh, use this new technology. Well, if it's for drug development, the drug developers need to want to use this technology and find it useful. And I encourage everyone to find out what are the problems, the questions of interest that a new technology could help solve. And if you can find questions that the regulators and drug developers are struggling with and a new technology could help solve, that might be a good place to focus. On that note, and what specific regulatory questions can new drug development tools help address today? Here is a image I've used a number of times. This came from a former staff member in the division that I led in the Center for Drugs, and represented drug development. You have a lot of compounds, your potential drugs you're assessing in drug discovery and early development, and you progress to an investigational new drug application, clinical studies, phase 1 and phase 2, phase 3 studies, eventually a marketing application with the hope of getting an approved drug. And with various new technologies and how you would translate those into regulatory use, these can have a big impact in drug discovery and early development. And you really can look at the problems and questions in the same way, but the context of use would be for a drug developer. And what are the questions they're facing in terms of advancing new compounds forward internally? At FDA, when we talk about qualifying new drug development tools for regulatory use, we're focusing on the regulated aspects of drug development. So starting with an investigational new drug application and progressing as clinical development continues, there can be applications for in vitro models to help answer questions that emerge during clinical development, in addition to informing when you're first moving into human studies at the investigational new drug stage. So on that note, this was the a part of the 2023 version talk. I think the goal of this workshop, as we put it together last year, was to bring all the stakeholders together. So it's a joint effort of figuring out among the different stakeholders the why, how, and what. And so thank you for joining. It's a pleasure to be back. And on the note of a updated version for this year, I did feed in the program from the web of this workshop, and this is what it came up with. So very creative. I still think it did better with the AI conference. There's a lot going on here, but and again, we need to define spelling out of the context of use of this model. But thank you very much.